Good evening, everyone. If I could just ask folks in the back to take a seat, and we're going to get started. Good evening, everyone. I am Mark Roithmare. I have the pleasure and honor of serving as the CEO of the Alzheimer's Drug Discovery Foundation, and I want to welcome all of you to our third in this annual science series that we have here in Palm Beach on behalf of the Alzheimer's Drug Discovery Foundation. Hats off to a couple who are sitting just in front of me, but a couple of rows back. Heidi and Tom McWilliams. This science series was their brainchild. We're in the second year of it, the third in this year's series. Heidi and Tom, thank you very much. Heidi, this may be the first one we've ever had an open bar during the whole thing, so this is going to be by far the most interesting, right, that we've had tonight. Heidi kind of double dips for us tonight, not only being the originator of this science series, but she's also the co-chair for the benefit this evening, along with Nancy Goods. Nancy, where are you? Where's Nancy? She's got to be in. There she, the grand entrance, Nancy Goods in purple in the back. You can't miss her. She was co-chair and founding chair. This evening. It will be our 6th annual event that we're having, Hope on the Horizon event. Also in the room with us tonight is the third of the co-chairs, Ms. Judy Glickman-Lauder. Judy, do you just want to give a wave? And all of that is, we have our fabulous honoree in the room, Bonnie Lautenberg. Special night tonight for many reasons. One of them is that we are beginning tonight to celebrate what is the 25th year or the 25th anniversary of the Alzheimer's Drug Discovery Foundation. Thank you to two incredibly handsome gentlemen who are brothers that are in the room tonight. Ronald and Leonard Lauder. Ronald's in the front, Leonard's in the back. Along with this fella to my right up on the stage, Howard Fillit, they founded the ADDF 25 years ago in 1998 with a couple of simple ideas. One, the ADDF would be a venture philanthropy. And what does that mean? We do not make grants to academic institutions or biotechs. We make investments in academics, in biotech, so that if the science pays off with any return on investment, the ADDF gets a part of that and plows it right back into the science. So that's number one. Number two, it would fund both academics and biotechs, and sometimes it would take an academic and spin it out to a biotech. Three, the organization would approach this through science, focusing on three things and three things only: prevention of Alzheimer's, the diagnostics of Alzheimer's, and some things you're going to hear about tonight that are related, called biomarkers, and therapies to attack Alzheimer's. A couple of claims to fame across all three of those. Diagnostics. There is a PET scan and there is a blood test that can tell you if you have Alzheimer's or if you are susceptible to Alzheimer's. That is brought to you by Dr. Howard Fillit, the Lauder family, and the ADDF. Now, many of you will say, well, why would I want to know? What could I do about it? We now know, because of the ADDF, you can actually prevent it. There are things you can do that will push out the onset of the disease. Could be 6 weeks, could be 6 months, could be 6 years, all right? And that's where the science is. And last but certainly not least, there are therapies. And you'll hear a little bit tonight, the first new therapies in 20 years that can actually do something about Alzheimer's are coming to the market. The reason they're coming to the market is because of these diagnostics and biomarkers that ADDF has brought to you, so that can say, this is what works, this is what doesn't work. And what's much more exciting than the 2 or 3 drugs that you're going to hear about that are just coming to the market is the cadre of drugs that's just beyond that, that's going to be these complementary or combined therapies. So you're going to hear a whole lot about that tonight. You're going to hear a whole lot about something called the biology of aging in the approach that the ADDF takes. And also, to my right, is Dr. Miranda Orr, who is the future of these new therapies that are coming. And so you're on cutting edge, really exciting news tonight. The last piece I need to do before I turn it over here is introduce Dr. Howard Fillit. This is the part that always makes Howard nervous, because I've got an open mic and he's not sure where I'm going to go with this. He is a physician, he is a scientist. He has spent the last 40-plus years dedicating what has actually been his entire life to understanding, combating, and putting forward effective treatments for Alzheimer's and its related dementias. He is brilliant. Any day, any time, any place, if you bring up anything that has to do with the science and the medicine and the approach and the policy to Alzheimer's, he can go deep, he can go wide, and he does it without a note. He's almost savant-like in these qualities. He also happens to be kind, some of the time, funny, most of the time, and he cares every minute of every day, and you'll see that come through. He's also somebody that, for myself and our board members here and committee members and the Lauder family, he's a good, and dear friend. I give you the remarkable leader in the field of science of Alzheimer's, Dr. Howard Fillit.
Well, thank you, Mark, and thanks to all of you for joining us today for our scientific symposium. As Mark mentioned, I've been doing this over 40 years, and I remember back 50 years ago, really, when I was in med school, Alzheimer's disease wasn't in my textbooks. I never heard the word. When I was training as an intern and a resident in hospitals in New York City, Mount Sinai in particular, during the mid-'70s, treating hundreds of people, elderly people, I never made the diagnosis of Alzheimer's disease. And it wasn't until 1980 when I walked into a nursing home on Long Island where I met my first patients, and I was told that these patients, my first patients with Alzheimer's disease, and I realized what an important problem this was going to be and how interesting it would be, what a challenge it would be to develop drugs, because this is a disease that uniquely and solely robs us of everything that we are as human beings. It robs our mind. And not only that, but it's a huge economic and social problem around the world now. I learned recently that in Australia, the leading cause of death among elderly women is Alzheimer's disease. And in most developed countries around the world, among the elderly, Alzheimer's disease and related dementias are the 5th leading cause of death. And I say that not just because of the economic issues. It's the most expensive disease in most developed countries, including the United States, almost $300 billion in direct and indirect medical costs, long-term care, lost productivity of caregivers, and so on. But it's also not only a chronic, progressive neurodegenerative disease, but it's a uniformly fatal illness. And so we really need to address new drugs. I started my lab and ran one of the first clinical trials in Alzheimer's at Rockefeller University back in 1981. And I give you that history because today you'd think, well, 40 years at this and we don't have a cure, but I've never been more excited about where we are than where we are today. We have over 140 drugs in development, which is huge. I remember when I started, there was basically nothing going on. And recently, you've read about it in the newspapers, we had the first drug approvals in over 20 years for Alzheimer's disease. Many failures, but we learned from every failure. And now we know how to do clinical trials. Imagine that in our field, we really didn't know how to do the clinical trials. And as Mark mentioned, we didn't have the biomarkers. Imagine doing a trial to prevent heart disease by reducing cholesterol if you didn't have a cholesterol biomarker. Imagine trying to develop a drug for hypertension if we didn't have a blood pressure cuff. But now we have those biomarkers for Alzheimer's disease. We developed the PET scan that led to the recent approval by the FDA. It's called an accelerated approval for 2 drugs, one from Eisai Biogen, and one which I believe will be approved in the near future, in the next quarter or 2, from Eli Lilly. And these drugs do slow down the rate of decline in people that have amyloid in their brain. And the reason that we could do that is because of the PET scan that our foundation helped to develop, which actually sees the amyloid, which is the characteristic feature in the brain of Alzheimer's disease. We can see it. I use it in my small clinical practice. I can send somebody over to the radiology office, and within 24 hours, I can tell them with about 95% certainty if they have Alzheimer's disease. We've learned from that the disease starts 20, 30 years before people develop symptoms, so we can identify and conduct prevention studies. And the FDA's approval on these 2 new drugs that are going to be coming to market was based primarily thus far on the fact that the drugs that are coming to market, I believe, completely clear the brain in about 80% of people from the pathology that is the amyloid. We're also invested in a new blood test that's on the market in 49 states. Imagine a situation which is possible now, it's present now, that you can go to the doctor with a memory complaint, and if you want to know if you have Alzheimer's disease, you can get a blood test. And that blood test correlates with the brain scan, so we know it's valid. And that's kind of part of the normal workflow of the way doctors work, the way you all expect to go to the doctor. You want to know, imagine there's a blood test now that can tell with 90% certainty if you have Alzheimer's disease. That's a revolution, and it's already being used in clinical trials to identify people who have Alzheimer's disease who are eligible for the trials. One phase 3 clinical trial in Alzheimer's disease, where you get proof of efficacy for the first time, costs $300 to $400 million. And you need 2 of those studies to get an FDA approval usually. And half of the cost of a clinical trial in Alzheimer's is finding the patients. The brain scan has been used previously to find the patients, but it costs somewhere between $5,000 and $8,000 per scan. So finding the patients costs half of that $400 million. With the blood test, we've reduced those costs by somewhere around 6 or 8-fold. So it's been a big advance, and it's already being used in clinical trials. Now, the other thing that's changed is that 7 years ago, 75% of the clinical trials that were being conducted around the world were directed against beta amyloid, which is this characteristic molecule in the plaques that are characteristic of Alzheimer's disease, and the tangles, which are the dying neurons. Today, 75% of the drugs in development are not beta amyloid or tangles-directed therapies, but therapies that are directed against innovative new targets like inflammation. Imagine that with Alzheimer's disease, there's inflammation in the brain, and that leads to faster progression. People can have amyloid in their brain, but if they don't have inflammation, they probably won't get symptomatic. There's also other aspects of what we call the biology of aging, and that's been our scientific strategy for many years. And there's about 8 different or 9 different pathways that I won't go into because it gets very technical, of the biology of aging that are now targets for the development of new drugs. And the reason that we focused on the biology of aging is going back a long ways, that aging, by far, is the leading risk factor for Alzheimer's disease. So you could ask the question, why is aging the leading risk factor for Alzheimer's disease? Well, it's a risk factor for many other diseases. Think about heart disease. Think about cancer. So all of these diseases of aging and old age increase due to the aging process. And so we've sort of characterized the aging process according to these 8 or 9 strategies, which I won't go into except for one. And the one that we want to talk about today is we're very privileged to have Dr. Miranda Orr with us. Dr. Orr is an assistant professor of gerontology and geriatric medicine at Wake Forest University. And her work is innovative in that she's really uniquely centered on the aging of cells in the body and how they're detrimental and contribute to inflammation in Alzheimer's disease. Miranda identified what are called senescent cells in the brain of patients with Alzheimer's disease. Now, this is important, and I'm not going to take away your thunder, Miranda, but I just want to introduce that this is really important because, as Miranda is going to tell you more about, these senescent aging cells, first of all, they're like cancer cells in that they don't die, and they cause inflammation. And so we're taking a risk on doing actually a clinical trial to remove senescent cells from the brain of people with Alzheimer's disease. And we've invested with Miranda at her university to conduct that trial using 2 drugs, which Miranda will tell you more about. And I just want to say this trial, which I hope we'll talk about, is representative not only of a novel pathway directly related to the biology of aging, but it's also an example of combination therapy. And as a concept, let's think about people that with hypertension or diabetes or cancer. People are on 2, 3, 4, 5 drugs at a time. That's because to control the cell, to control the cancer cell, to kill the cancer cell, it has many ways of getting around a single drug. And so you need to attack 2 or 3 or 4 pathways in the cancer cell in order to effectively remove the cancer. You need 2 or 3 different ways to control glucose in diabetics or to control hypertension in people with high blood pressure, because we have to get this combination therapy to attack multiple pathways in the cell. That's one concept that's illustrated by Miranda's clinical trial. And the other one I want to mention is called repurposing. Imagine thalidomide back in the 50s, horrible drug for people, for pregnant women. It was prescribed for anxiety. Horrible babies were born. The drug was taken off the market. About 10, 20 years later, a doctor thought that thalidomide might be good for multiple myeloma. And so he did clinical trials repurposing thalidomide for multiple myeloma. Today, the leading drug for multiple myeloma is thalidomide or a derivative of that. And there are many, many examples of that. And what we've done and what Miranda's going to tell you is repurposing a cancer drug for the treatment of Alzheimer's disease. So another concept, because the advantage of repurposing is that we know the safety profile of these FDA-approved drugs, we know their mechanism of action, and so we can intelligently try to repurpose drugs from other diseases for the treatment of Alzheimer's. And our foundation has funded about 19 different medications from Parkinson's disease, from ALS, from diabetes, from hypertension, to treat people with Alzheimer's disease. And doctors can prescribe these drugs off-label if they want, and there's other ways that we can commercialize these drugs the same way that thalidomide was commercialized for multiple myeloma, and that's one of our efforts, is to take advantage of positive studies which we've reported, which our scientists have reported in the medical literature, and figure out a commercial strategy for bringing those drugs to market. So with that, I want to turn it over to my colleague and one of our funded scientists, Dr. Miranda Orr.
Thank you so much, Howard. And I want to start by thanking the Lauders, Mark, Howard, ADDF, and Nancy for supporting researchers like myself. We're very dedicated in this endeavor, and the reason I'm here is probably the same reason that many of you are here. I lost somebody very important to me to this terrible disease, and that was my maternal grandmother. My grandma did everything right in life. She graduated her high school class valedictorian. Shortly after she graduated, she met and married my grandfather. They bought a farm outside of a small town in northeastern Montana, which is where I'm from. They had 6 children. They raised their family, and they did everything right. So we don't have a Mediterranean diet up in Montana, but but my grandma did eat healthy. She raised a very large vegetable garden, and she was able to feed the family in the summer, and she produced enough to can or freeze to feed the family in the winter, and they raised their own produce. So good diet, as good as we can up in Montana. Exercise. She was a farmer's wife. There was a lot of manual labor, not necessarily all of that manual labor is good wear and tear, but she was outside, got a lot of vitamin D, a lot of exercise. Check. She had a very good social network. She loved to go into town. She loved country dancing with my grandpa. She was involved in several different charities and communities in town. She was very involved in my life and in the lives of my siblings and my cousins, so very socially engaged. And she kept her brain very active. Some of my earliest and best memories of my grandma are playing cards. I tell people she actually taught me how to remember. One of my earliest memories of my grandma was sitting on her lap, and during Christmas breaks I would stay with her, and we would have just a normal deck of cards, and they'd all be face down, and I would choose two, and if they didn't match I had to put them back over, and then she would choose two. If they didn't match, put them back over, and we kept going until we had a match. If you got a match, you got to keep them and keep going, and the winner was the person with the biggest stack at the end. And Grandma instilled a lot of confidence in me, and I think she liked to tease my brother that I had a better memory than him. He was older than me. So it's quite ironic that in high school she started losing her memory, and she started losing interest in a lot of the games that she had taught us to play, and it didn't get better. She got the diagnosis of Alzheimer's disease, and she passed away the year that I graduated from college. And as a very young, naive 20-something, I decided I should cure this disease. Not that a lot of people hadn't been trying. But would be a fourth-generation farmer. I didn't know what that meant. I didn't know how you cured a disease. I didn't know any scientists, but I talked to people. I got a lot of advice, and I found people that not only supported me, but had a similar mission. And so I applied to one graduate school, and that was at Montana State. I got my PhD there and learned how to study Alzheimer's disease and stem cells in mice. And then for my postdoctoral fellowship, which is the next stage as a scientist before you make it in the real world and develop your own independent lab, I went to San Antonio, where my husband at the time had a job, and I had the privilege of training at one of the world premier institutes that focuses solely on the biology of aging. And this was revolutionary to me. I didn't know we could do anything about aging. I thought it just happened to us, but it turns out that it doesn't just happen to us. We understand a lot about what happens in our cells as we get older, and the great news is if you intervene on them properly, you can delay aging, you can delay disease, you can extend healthy lifespan. This was incredible to me because the number one risk factor, as Howard said, for getting Alzheimer's disease is aging. So I thought maybe we could use some of these hints to treat aging. And as a postdoctoral fellow, we discovered, as Howard said, a fundamental aging process that contributed to Alzheimer's disease. We treated mice with drugs that cleared these senescent cells. They're sometimes called toxic zombies, and I'll get into that a little bit more. We saw improvements in the learning and memory of the mice, and colleagues in my field had already tested these drugs in people with different conditions, because as Howard said, all of these aging processes lead to a whole litany of diseases. So my colleagues had already tested it in chronic kidney disease. They had already tested it in idiopathic pulmonary fibrosis, so we knew they were safe. So with that information, I submitted an application to the ADDF, and they funded our trial, and we are currently conducting the first phase 2 trial, clearing these aging cells from people with amnestic MCI or early Alzheimer's disease. And something really incredible happened to me in my career, and that is the National Geographic became very interested in our study. They highlighted some of our work in the January 2023 issue, and they're going to be highlighting the study that ADDF is funding in the September issue. So keep your eyes out for that issue, and they actually were in their lab visiting us at Wake Forest when our very first clinical trial participant completed the treatment stage of the trial. And so they have a picture of that individual going through that stage.
So Miranda, what drugs are you testing in this clinical trial that are anti-aging drugs? Could you tell us how they work and why you picked that combination?
Yeah, it's dasatinib, which is a broad-spectrum tyrosine kinase inhibitor. That might not mean anything to you, but it's FDA approved to treat leukemia. So there's a lot of information about this drug, safety profile, and that is in combination with quercetin, which is a flavonoid, and quercetin is in a lot of fruits and vegetables. You probably are getting a healthy dose of it just by eating a healthy diet. And so the reason why we chose these two the combination of these two drugs is they had been shown to clear the most types of aging cells across the body, and importantly, cross the blood-brain barrier. So a lot of drugs, they might do great things for peripheral tissues below the neck, but if they can't get into the brain, that poses a challenge. So we knew that they crossed the blood-brain barrier, and they had worked very well in our mouse model.
So the tangles that are characteristic of Alzheimer's disease, are they senescent cells, and do these drugs try to prevent the tangles and the neuronal dysfunction that occurs in the disease?
Yeah, so that was one of the breakthroughs early on, and that is, so in the brain, arguably the cells that have the most stress don't die. And that was something that I learned about in graduate school, and I had a hard time wrapping my head around that, because if you look at these cells from a person that passed away with Alzheimer's disease, you're looking at their tissue under the microscope, and they look very stressed, but we know they don't die, but they are in the right place and right time. So what that means is the more tangles there are in the brain, the worse the dementia, the more healthy brain cells die, and that's what senescent cells do. They don't die. They actually can't die. They figure out a way to survive with damage. They release molecules to tell the immune system, get me out of here. But if the immune system doesn't hear that signal, the cells keep sounding the alarm, and this leads to inflammation. And that causes damage to healthy cells, and those cells can either die or become senescent. And it is the cells with the tangles.
So how does dasatinib kill those cells?
Yeah, the way that these cells figure out how to survive, dasatinib actually goes in and takes out that armor. So they're no longer able to survive. So essentially what these drugs do is they let the cell kill themselves, because they're simultaneously trying to die. So if you remove their ability to live, they're cleared.
Well, why do you need the quercetin in combination then with the dasatinib?
They work synergistically, so dasatinib alone can clear some senescent cells in some tissues, but when paired with quercetin, you get a broader clearance of senescent cells, which we think is really important. As we get older, we're accumulating all of these cells in our tissues, and people with Alzheimer's disease don't only have Alzheimer's disease. They often are suffering from diabetes, arthritis, all of these other diseases that senescent cells contribute to. So strategically, if we're able to, with a single strategy, clear out all of these bad guys, we think that's going to give people the most benefit.
Well, dasatinib is a cancer chemotherapy drug, and most of those are associated with very serious side effects, but since cancer is such a serious disease, not that Alzheimer's isn't, and I think we need to talk about that, but obviously also a fatal disease. But how do you dose dasatinib differently in Alzheimer patients than you do in cancer patients, especially since the Alzheimer patient is typically a frail elderly person who would be subject to these side effects?
This is such a great question, and one of the beautiful innovations of this strategy. So we have a couple differences that are key differences from cancer treatment. So one, the dose is lower, and two, we give it intermittently. And the reason for that is you don't have to keep dosing if the cells aren't there. So we dose 2 consecutive days. The drugs let the cells kill themselves, and you don't need to give it again until the senescent cells come back. So we only give it 2 days, and then the patient or the participants have 2 weeks off. We repeat that 6 times. So in the period of 3 months, they're only getting the drug 12 times. And then we have an extended follow-up period where we bring the participants back every 3 months. They're not on placebo or dasatinib and quercetin, and we take their blood and measure their cognition. And the goal of that extended follow-up period is to determine if and when senescent cells come back. It might turn out that you only have to take these drugs once every 3 months, once every 6 months, maybe once a year. Just clear out the source of inflammation, and then you don't take them again until they reappear.
So we talked a lot about biomarkers in the introduction. What biomarkers are you using in this trial to see if the drug is working?
The primary biomarker that we're looking at is senescent immune cells. And there's been a lot of work in that area, not only from our group, but from others in the field, showing that when the immune cells become senescent, that becomes a huge problem because you're accumulating senescent cells in your tissue, and then your immune system actually can't clear them.
So are there blood tests like beta amyloid in the blood or spinal fluid tests or neuroimaging that you're using to see what the effect of dasatinib and quercetinib is in the clinical trial?
We are. We're looking at several different biomarkers in the blood and in the spinal fluid. We're also using a lot of different brain imaging techniques, including tau PET, because we do know that the tangles are a source of senescent cells, so we're looking at a reduction in that before and after treatment. And one of my colleagues at Wake Forest is actually helping me build a new tracer to identify senescent cells, so we would be able to image them in patients.
So tau imaging is on the market actually at this point, and it's quite interesting because it correlates with cognition and it's a measure of the dying cells. So you're saying that the tau imaging that's out there now, mostly from Lilly, I guess, is the main agent, is able to actually, in fact, is neuroimaging senescent cells in the brain.
We do think this is the case. We also, however, do know that not all cells with tangles are senescent. So we're thinking about a combination imaging strategy where we use the existing tau PET tracers and then have another one that is really specific to senescent cells.
Mm-hmm. And what patient population in particular are you studying with Alzheimer's, like MCI, early dementia, which?
Amnestic mild cognitive impairment in early Alzheimer's disease, and everybody is age 60 and older.
Mm-hmm. Okay. And are there any other entry criteria that you have for this?
Yeah, so there are quite a few different inclusion and exclusion criteria. Of course, no other pre-existing or chronic diseases or a history of cancer in the past 3 years, because we are using a repurposed chemotherapy. We do take EKGs every study visit because one of the known side effects of dasatinib can be some changes in heart function. So we look very carefully at that. But what I can say is, a lot of participants, when they're interested in this new innovative trial, they do get a little bit nervous when they hear the word chemotherapy. But our dosing strategy, along with the lower dose, the intermittent dosing, it's really, we have not had issues with safety.
So how many people will you enroll in the study when the enrollment is complete? How many people do you hope to enroll?
A total of 48, and they will be equally randomized to either placebo or the active study arm, and we're about 1/3 way through right now.
So this would be what you'd call a phase 2A study, I suppose, actually quite a small study. I believe we gave you $3 million, if I remember correctly, for about 36 months by the time you enroll everybody, and then I think the treatment period is 1 year, is that right?
It's 3 months, but then the whole enrollment period is 1 year because they remain enrolled for an additional 9 months for the follow-up.
Mm-hmm. So with $3 million, you're saying we probably, it's powered, the study is powered enough for $3 million and 48 patients to get an answer. Is that what you're saying?
We will know whether or not we are on the right track and can move to a larger study.
So this kind of study is what we call an exploratory study. It's asking a question to see if there's a signal, and if there's a signal there that looks good on biomarkers, probably, because the study is even more underpowered for clinical outcomes, right? But if there's a signal there, then hopefully what we'll be able to do is figure out the next step, maybe get $30 million or $50 million to do the next trial, which would be maybe a few hundred people to get a really good answer. And the other thing you'll get out of the phase 2A is safety. Can these drugs be used safely in elderly people with Alzheimer's disease? I don't know if you want to comment on that.
Yeah, I agree. Yeah.
Okay,
Howard,
great.
is that?
Now, just to change the subject for a second, inflammation is a hallmark of aging, right? What about other methods for reducing inflammation in the brains of people with Alzheimer's disease? I mean, we know from studies that have been done that at autopsy, for example, and actually Alzheimer described in 1906, when he described the first case, he described that there was inflammation around the plaques. And in my lab back 30 years ago at Mount Sinai and so on, and other data has shown that when some people do not mount an inflammatory response in the brain against these plaques, and they don't have symptoms and they don't progress, And some people, most people, develop an inflammatory response, and it seems like the inflammation is actually causing the progression of the disease as much as the sort of inert, if you will, amyloid plaques. What other approaches, based on the biology of aging or whatever, would you talk about that would be sort of an anti-inflammatory approach to what's going on in the brain, particularly against these inflammation cells, which are called microglia? And if you might, that's a technical term, I know, but.
So the beauty of this treatment is they also clear dysfunctional microglia, which are a major source of inflammation. And we are starting to think about even an additional combination therapy and what sort of anti-inflammatory would be best to use, because while senescent cells are a primary contributor of inflammation, we don't think that they are the only contributor of inflammation. So it might require an anti-inflammatory as well.
So that's a brief summary of what what Miranda's doing, and there's 140 other clinical trials going on right now in our field addressing a whole host of maybe at least 8 or 10 other mechanisms that are novel and don't involve this amyloid. So we're excited about the amyloid monoclonal antibodies coming to market. It's an incredible first step. It's not a cure. It slows the disease down by somewhere between 25 and 35%. We want to get to 100%, slowing it down, and ultimately to preventing the disease. And you know, for the first time, we're really, I think, on the right path. So thank you, Miranda. We'll open it up to...
We're going to open it up to questions now. I just want to pre-warn our presenters that there has been this open bar, and we've done a lottery, and we've picked the first person for the question, and it happens to be Ronald Lauder. So, Ronald.
Thank you, and thank you, Miranda, and thank you, Howard. Frankly, I've been fascinated by the idea with biomarkers. We can tell if somebody at the age of 30 and 40 is a candidate for Alzheimer's. And we are working on things now that could, in fact, be given as an antibody to Alzheimer's. I've always been fascinated by Dr. Jonas Salk, where about polio, they couldn't find a good cure for polio, but they did find a type of injection you can get to stop polio, people having it. My question is, how close are we to finding a cure for Alzheimer's? Not a cure, but to prevent Alzheimer's.
So there was just a very large study of thousands of people that was conducted by Eli Lilly, because as you said, Ronald, we can do these brain scans on people in their 30s and 40s and detect people that have Alzheimer's, just like you could get a coronary angiogram when you're 40, and the cardiologist will tell you have a plaque, but you might not get a heart attack till you're 70. Same thing. We can detect the amyloid in the brain now. We know the disease starts at least 20 years before people develop symptoms. So a study went out there that screened the community with these brain scans, found a whole bunch of people that had amyloid in their brain and they had no symptoms, and then treated them with one of these monoclonal antibodies to see if they could prevent cognitive decline and prevent the disease from occurring. And one of the things that we've learned from a study that was done by a guy named Brookmeyer a number of years ago was that if we could just delay the onset of Alzheimer's disease by 5 years, we would reduce the number of people suffering from dementia by 50%. And that would be a huge impact, and I believe that's an achievable goal. The average age of onset of Alzheimer's is about 75, and the average age of death is about 80. And as geriatricians and people interested in healthy aging, delaying the onset till the age of death from cardiovascular disease, I think we'd all like to die in the middle of the night from a heart attack rather than losing our minds over 7 to 12 to 15 years. Anyway, this study just reported out last week on the prevention, which is exactly the first of its kind that you're referring to, Ronald. And unfortunately, that study failed. In other words, they were able to show that there was a decline in the amount of amyloid in the brain, but it didn't seem to have an effect in delaying the onset. And I think the problem with that particular study was that that was a previously failed antibody in Alzheimer's patients that had dementia. but we're going to see other antibodies and other treatments against the amyloid that will come out in other prevention trials. And so all over the world now, there's prevention trials going on. And let me just say one more thing. We are funding another geriatrician from Finland named Mia Kivipelto, who's also one of our board members now. And Miranda talked about all these prevention methods, and Mark spoke about exercise, sleep, diet, stress, caffeine, cough, smoking, alcohol, depression. Okay, so all these things, but they've all come from what we call epidemiological studies. What Mia did was the first randomized clinical trial of prevention in the history of the world. And she put one group to get an intensive prevention regimen with all these factors in play, and another group that just got advice on the first visit and then were left to do on their own. And what she showed was an intensive prevention regimen in a randomized trial could slow the rate of onset of cognitive decline in elderly people. So that would be like doing a lifestyle intervention and managing your diabetes and hypertension, which are the main comorbidities for Alzheimer's disease risk factors. In heart disease, very similar. The only difference is in the prevention of Alzheimer's, we say, as you said, we want to maintain our social engagement and keep our minds active because the brain is like a muscle. But what we're doing, so that study is being replicated now in 45 countries around the world. And 45 different countries want to know if this really works. Now we're doing the 2.0 version with Mia. Just like in the prevention of heart disease, you do lifestyle management, you do comorbidity management of diabetes and hypertension and atherosclerosis, high cholesterol, but you add a statin. And so the drug we're adding to her randomized trial is metformin, which is repurposing of a drug for diabetes, the leading diabetes drug in the world, but has an effect on the aging brain. We funded a study at Columbia University in New York of metformin for the treatment of Alzheimer's disease with about $1.5 million. About 5 or 6 years ago, that investigator just got $40 million from the National Institute on Aging to really look at whether metformin, which is the leading anti-aging drug in the world, I think it's fair to say, you might want to comment on this, Miranda. So we're adding on the leading anti-aging drug to the lifestyle intervention and seeing if that will make it even better, that we can prevent Alzheimer's disease. So there's a lot of excitement about prevention of Alzheimer's, and we're really excited about it. Did you want to comment?
I don't have a lot to add, but metformin is an example of a compound that really does target aging, and it targets so many different aspects of aging. Very well studied, very good safety profile, and very inexpensive.
I've enjoyed this very much, and how do you become part of the study group?
Call me. So we are currently recruiting from North Carolina, Wake Forest, so Winston-Salem, and San Antonio. We are looking to add additional sites. So given the number of study visits, you do need to be located in those regions at this point.
I think we can agree that the last bastion for us to conquer is the brain. Parkinson's, dementia, Alzheimer's, put it all together. I guess my question is, there's very little funding that's come from the venture capital community. What do you think of the steps that you have to take and the progress you have to make before that happens? Because there's obviously an onslaught of capital come from those sources. And a parenthetical question is, to what extent is artificial intelligence helping along the way?
Well, to answer your first question, we are venture philanthropy. So in 1998, our incorporation papers to the IRS said we were going to do just really one thing, which was accelerate the development of new drugs with biomarkers, with clinical trials, with preclinical drug development. But we also told the IRS that we were going to be investing in biotech companies. And so far, we've made 170 investments in biotech companies around the world, not just in the United States. And this is a really high-risk endeavor. Over 90% of drug development programs fail, and especially in Alzheimer's disease. We had a drug approval in 2003, and it's 20 years later, and we finally got a tentative approval in our field. So for investors, with a long time frame and a different kind of risk tolerance, this is not a happy field. When I started, when the foundation started, I did a survey of the biotech and pharma industry to see how many companies there were that were actually having programs in Alzheimer's disease in industry at that time in 1998. At that time, there were 1,600 biotech and pharma companies in the United States, and only 3 had any kind of program in drug development and Alzheimer's. And that's changed a lot, but I think venture capital is getting more confident with the knowledge that we've gained over the last 40 years. They've gained some confidence in our ability to do clinical trials, just like if you read The Emperor of All Maladies, the cancer field and the NCI had to figure out how you do cancer trials. We've learned how to do Alzheimer trials, and I think we're starting to see more confidence in the investor world for appetite for Alzheimer's because the risk has been mitigated to a certain degree. I think the philanthropy plays a very important role in mitigating the risk, and I'll say without being self-promoting and with all honesty that we hear from many investors that they want the ADDF approval. They want the endorsement from ADDF because we're a nonprofit and we have an excellent reputation for doing our due diligence. We get calls all the time from venture capital that don't have experts of Alzheimer's disease on staff. If we fund something, there are companies that tell us they don't necessarily need our money, but they want our endorsement. And I think that's another way that VCs are getting some confidence. I don't want to push that too hard because it's self-promoting, but it's true.
So Howard, we lived that. I have Jupiter Neurosciences as we are into Alzheimer's and Parkinson's disease. And you're absolutely right. Your comments about the VCs and so on. Started this company 7 years ago after my mother passed away in Alzheimer's. And VCs were then Alzheimer's? No, that's like 20 years out. Why should we invest in you? So what you're doing here is fantastic, and you actually are. Now they're starting to ask us, okay, how would the ADDF say about it, and so on. So thanks a lot for what you're doing.
And let me say, just so people understand how we do operate like a venture capital group. We do due diligence. We have a chief financial officer who is formerly an investment banker. We have a business review board of venture capitalists and investment bankers and people like that. So we do very serious due diligence. And I have 11 neuroscientists on my staff. I mean, this is not me sitting around a desk. We have 11 neuroscientists, day in and day out. They're searching the world for new opportunities, and they're doing due diligence on these companies, very extensive due diligence before we decide to invest. And we've made, as I mentioned earlier, over 170 biotech investments in the last 25 years. Ginger.
You've spoken about multi-purposing drugs. In diabetes, that's thrown off Ozempic and metformin. Is there something about the nature of diabetes that has been permitting these drugs to be spun off?
Yes, exactly. But before I answer that, I want to turn it to Miranda because she and I talked about insulin resistance in the brain. And let me just say, the brain is 2 or 3% of the body weight. But at any given moment, it uses 20 to 25% of the body's energy, and most of that energy comes from glucose. And so when the brain can't use glucose through insulin resistance, and I'm stealing your thunder, I'm sorry, there's neurodegeneration. So I'll leave it at that.
Yeah, so we find that these senescent cells, these toxic zombies, they're actually insulin resistant. Just like muscle cells or fat cells in other tissues that lead to diabetes. And so a really interesting area that we're looking at is combination therapy with these drugs that clear senescent cells but are also traditionally used to treat diabetes. Diabetes alone significantly increases your risk for developing Alzheimer's disease. So there is a really close connection there.
So we funded an investigator at the Imperial College of London several years ago to study one of the leading diabetes drugs. It's called liraglutide or Victoza. And it's kind of like metformin, but it's probably better for diabetes in some ways. And it was, again, an exploratory study, a small study to see if this diabetes drug could benefit patients with Alzheimer's disease, even though they weren't diabetic. Just to see, because we all develop a certain degree of insulin resistance with aging, and the brain does. And there were positive outcomes from that small study. And so Novo Nordisk, which participated in the study and is the pharmaceutical manufacturer of liraglutide, has Ozempic, which is the next generation of these drugs called GLP-1 agonists. And now, because they saw the data from the small study, by the way, we committed $750,000 to that study. General Electric donated the brain scans that were necessary for the study. Novo Nordisk developed the drug, and it ultimately became about a $7 million study from the seed money that we put into the Imperial College of London. But once Novo Nordisk saw that data, they embarked on a 3,000-patient study of Ozempic, of semaglutide, for Alzheimer's disease, and that study is going on now. Thank you. Any other questions?
Yes. Oh,
Yes, please.
I don't need a mic. Is there any correlation between plaque in the heart and plaque in the brain?
No. I mean, the question is there any correlation between plaque in the heart and plaque in the brain? They're two totally different things. The plaque in the blood vessels in the heart is cholesterol, and it's causing blockage and has other proteins in it. The plaque in the brain is mostly composed of this protein called beta amyloid. But to answer your question in a slightly different way, is heart disease a risk factor for Alzheimer's disease? And you know, since we're two geriatricians here, we think of the brain as part of the body, but neurologists don't, okay? No, and I say that with all seriousness because as a geriatrician, I thought of aging as from a holistic point of view and looking at these risk factors. But part of what I think hampered the field for a number of years was not thinking about systemic risk factors as risk factors for Alzheimer's disease. So things like atrial fibrillation, where you're throwing up little clots all the time, or ischemia from heart disease, or blood vessels in the brain that are not perfusing the brain well enough, are risk factors for Alzheimer's, and even vascular dementia, which is another form of dementia. 50% of people in the community, living in the community, actually have what we call a mixed dementia, which means they have pathology of Alzheimer's with plaques and tangles, but they also have vascular disease. So the real answer to your question is both, in terms of what's happening. The pathology is different, but what's happening is the two diseases, heart disease, vascular disease, and Alzheimer's are intimately related. I don't know if you want to add to that.
Yeah, I'll just add one quick thing. So Howard brings this topic up, neurology versus gerontology, and it is such a huge issue in our field. When I put my proposal in to study these drugs to treat Alzheimer's disease, we put outcomes in, meaning we wanted to measure things that just generally told us about a person's health and their aging. And reviews that I got back from my colleagues in neuroscience said, what in the world does walking speed have anything to do with Alzheimer's? And to me, it was just so interesting to get that comment, because people with Alzheimer's disease aren't just a head with Alzheimer's disease. It's attached to the rest of the body, and if you can improve any aspect of their well-being, it's a huge step forward, and it's thanks to ADDF for actually funding these types of studies so we get more information about the whole person.
And let me say, as people who've studied gerontology, and I actually edit the leading textbook on geriatric medicine and gerontology. About this much of the textbook is on gerontology, which is the study of aging, that this was begun about over 100 years ago. So like I said, we didn't really start doing research on Alzheimer's till about 1980 and 1981, but the science of aging has been going on for well over 120 years. The Journal of Gerontology was started in 1948. So there's a tremendous amount of knowledge about the aging process that hasn't even been considered as knowledge that we could use to treat Alzheimer's, and I think that's what Miranda and I are trying to do.
We've got time for two more, one's here and one's here, and
Okay,
then they're
good.
around if you want to talk to them after, so go ahead.
Hey, thank you so much. I just retired from 51 years in practice, and I've watched all these things. We didn't have MRIs or CAT scans or anything, and the progression has been great. I want to thank Bob for inviting me. He's with LifeWorks and has taken care of a number of my patients over time. This has not only been of interest, but I had two questions. One of them was about metformin, that already was asked. And the second one is about autophagy and or trying to clean up our own cells, partly with intermittent fasting. Can you speak to that at all? That's no big cure, but certainly we're all trying to do small things to make a difference.
Well, let me translate your question, and I'll turn it to Miranda. So the doctor is speaking about a process called autophagy, which in Greek or Latin, I don't know, it means self-eating. But the point is that as we age, our proteins, every minute of every day, our proteins are misfolding. Now, what does that mean? Has anyone ever cooked an egg? Do you start out with an egg that's going to become a chicken? And all of the proteins in the egg are functional because they're in their native function, and proteins have to be in their native configuration in order to function. And you throw that egg on a hot plate, and what happens? It becomes a scrambled egg. That's happening in every cell in your body every minute of the day because the tendency of proteins to misfold is great. And there's tremendous, through evolution, the body and the cells have have evolved tremendous mechanisms for managing these misfolded proteins. Because when proteins misfold, they become toxic. And that's what the beta amyloid is. It's a misfolded protein. Beta amyloid has a normal function in the brain. We don't know exactly what it is. The tau that causes the tangles has a normal function in the brain. But when they misfold, and the process in the cell to overcome the misfolding fails or gets overwhelmed with aging, these misfolded proteins that are toxic accumulate, they develop into plaques, and they cause neurotoxicity. So that's what the gentleman is asking. It's been a really hard target to drug, though. We have a lot of knowledge about the biology, but a really hard target to drug. And we're working right now with a company that's actually addressing autophagy as a therapeutic outcome. Maybe you want to comment, please?
Yeah, so it is a hallmark of aging, and so rapamycin, I don't know if the ADDF is funding any of those trials, but there are other drugs that do target autophagy to try to increase the clearance of these misfolded proteins.
Yeah. And I think it's, okay, one more question.
Just one more. In terms of NPH, have you found in your research that the senescent cells cause ventricular hypertrophy or enlargement in the cadaver studies?
In the heart?
No, in the brain.
So we have looked for senescent cells in a lot of different cell types in the brain. We do find that cells lining the vasculature do become senescent, but interestingly, they're in healthy people. So we think that this might be a natural process of those cells, that they become senescent and then they're cleared during a normal process. We have a study ongoing where we are looking at postmortem human brains from the age of 20, people that passed away at age 20, all the way to 100 plus, and looking for when senescent cells first appear, what cell types they are, and if their profile changes with aging. So we're finding bad senescent cells already in 40-year-old brains. We find good senescent cells in 20-year-old brains. So there's a lot of research effort trying to figure out the good senescent cells versus the bad senescent cells. Evolution didn't give us senescence only to be a bad guy. So it's an active area of research.
And Howard, you get the last word tonight, so anything you want to say, Howard, it's all yours.
Do I have 10 or 15 minutes?
No, you have 3.
Well, I just want to thank everybody for paying attention and listening and for all your great questions.
If I could, before we leave, just one or two things. A quick anecdote that I think will sum up a lot of what you've heard. You can look this up. There's a gentleman by the name of Dr. Mark Minton. He is the head of neuroscience at Eli Lilly. You can Google it. You can look him up. He's a real person. They have a drug that you heard about tonight that's about to report out in about 2 months, and the expectation is it will probably be, we believe it will be, once the data is revealed, the best of these anti-amyloid drugs. And Howard and I had a Zoom call with him, and the reason we had a Zoom call was to talk about the diagnostic that they're using in comparison with that, because we've done these diagnostics with Lilly, and when he came on, he said, "Oh, Howard, I want to congratulate you in the ADDF." And Howard said, "Well, geez, why?" And he goes, shouldn't I be congratulating you? And she goes, "Well, it's too early to congratulate us on this drug, but a lot of pressure, but we feel a lot of great hope, and quite frankly, this will be my life's work with this drug at Lilly, where he works as the head of neuroscience. You can't get a bigger job in neuroscience or Alzheimer's or Parkinson's there. And his whole purpose is to bring the drug to market." He goes, "Oh, well, I know that if this drug is successful, 35% effective. But good God, Howard, you were for 15 years in the ADDF, have been laying the groundwork for all the other drugs that are going to be used in combination therapies with this drug. So I'm congratulating you because you not only seeded the field, you not only developed the field, but you will now, in the next 10 to 15 years, complete the field." And this is what you've heard tonight. The beginning of that you've heard tonight, all right? Final commercial. We have a great night to enjoy, but we're not carpetbaggers from this ADDF. We're going to be back here on April 4th, the McWilliams Science Series. We're going to have, you've heard about her tonight, Dr. Mia Kivibelto, the top prevention scientist of Alzheimer's in the world. If you want to learn how to push it out, to keep it away from you, you want to be at the Norton April 4th for Dr. Kivibelto. And now, across the hallway, we're going to start our dinner. Thank you all. And thank you to our presenters.